going today? Let me go to this other screen. So where we're at is it's Tuesday, 4/7. I'm obviously kind of pre-recording this. Um, so yesterday we met, got your constant questions in yesterday. We met in the Teams Meet Now feature. I'm going to try to kind of keep in that habit, I think, kind of moving forward, at least I think right now, where, the beginning of class, I'm going to hit that Meet Now button in the team site, and you guys will roll on into class, and we'll talk about some things. You guys just got done taking your quiz, so hopefully that went well, and we will go from there. So today we're out here on Tuesday, going through the first part. Obviously, I will send you this link. I'm making the link right now for today's video lecture on beginning of Mendelian genetics. Tomorrow, then the second kind of portion of this lecture, and I'm imagining tomorrow will be a little bit shorter because my idea is we're going to probably finish up kind of the PowerPoint file today, and then focus tomorrow on Punnett squares and these things called pedigree charts. And that'll be sent as well later on. And then don't forget Thursday, the optional assignment is due, and then we'll have our kind of next team meeting on that day on Thursday as well too. So let's get right into it and flip to today's presentation. So just as kind of a reference point to you guys, chapter 10 is where we're at in the book, primarily sections 10.1 and 10.2. The second part of chapter 10 is a little bit more geared toward, the second part of chapter 10, section 10.2. This is more kind of the Wednesday's stuff, tomorrow's stuff, looking at Punnett squares and pedigrees. Today we're kind of setting up just a little bit about who Gregor Mendel was, and sort of the concepts that he contributed to what Mendelian genetics is. So that's kind of where we're at today, primarily in sections 10.1 and kind of the first section, or first half, I should say, of 10.2. So go ahead and flip to this next slide. So Gregor Mendel, he's pretty interesting. I got a couple of links right here, and we're going to actually go to the one link right now on the biography site, which is kind of cool. So Gregor Mendel is kind of a cool story. And we're not going to sit here and read through the whole thing by any means, but he was an Austrian monk, and he discovered the basic principles of heredity through really experiments in his garden. Isn't that kind of cool? So he kind of grew up in a rural community, not of any real high means or anything like that. Made his way into working at a monastery in Austria. And lived kind of this solitary life in a sense as an Austrian monk and living in the monastery, and they end up putting him, he becomes fairly renowned for his skills in mathematics and science, and he ends up being put in charge of the garden and making some experimentation. He was very fascinated by how certain plants ended up showing different characteristics. And he sort of set about trying to find a way to be able to mathematically explain that. Really, much of kind of what he did was, a perfect example of sort of the scientific method. He saw things happening and saw that there might be some reasonable explanation for that. He tested it, was very meticulous in his testing method. And very meticulous in his record keeping, and ultimately was able to assemble these sort of basic laws or scaffolding pieces that really govern modern genetics to this very day. He was born in 1822. That's a long time ago, right, guys? And he, did his primary experiments, I believe it was kind of like the 1850s. Right here around 1854, began to research transmission of hereditary traits in plants and plant hybrids. So, we're talking, 1850s. And what comes out of this are these basic laws that we're going to talk about today that really are still, guiding principles for how we understand genetics and heredity and how different characteristics are passed from one generation to the next. So it's pretty cool when you think about, this monk meticulously moving pollen from one pea plant to the next, um, in the 1850s, and tracking what he did to the point where he was able to mathematically sort of figure out what lays the groundwork for modern genetics. And I think we can all agree that really genetics has become really this wide open, amazing field in terms of how we understand how different characteristics are passed on from one generation to the next. And we're going to get a lot more into that in this unit. It's pretty cool. And it's really one of the cutting edge fields in biology right now at this very moment. All right, so here he is, Gregor Mendel, father of modern genetics. He used crossbreeding of pea plants. He worked with pea plants primarily to discover inheritance patterns and offspring. So what he would do is he would isolate and develop true breeding sort of series of plants. That meaning, what does that mean? It means that true breeding would mean that they would only have a certain characteristic generation after generation after generation. Excuse me. So he went through the work of really separating these pea plants to only show a certain characteristic. So then he could guarantee that only they could pass on that particular characteristic. So I'm going to kind of flip to this next slide. So Mendel crossbred true breeding plants with different characteristics, page 209 in your textbook. And I would suggest to open up, pause this right now and open up and go to this page in your textbook. So this is the exact image from page 209 in your textbook. So these parental generations. So he had gone through the meticulous duty of isolating plants that showed different characteristics for flower color, flower position, seed color, seed shape, pod color, and stem length. And he would isolate these plants and breed these plants and make sure that he would have true breeding plants that would have only purple colored flowers or white colored flowers. Axial location of the flower, which is kind of off to the side, or terminal, which is at the top or the end. Plants that would have either yellow pea seed colors or green, round, wrinkled. You guys kind of get the idea. So he would have these true breeding plants. So let's kind of maybe look at the purple versus white color example. These true breeding parental plants, and then he would meticulously cross pollinate these purple with white colored plants, white flower colored plants, and ultimately would see these pretty consistent results across, not pretty consistent, they were consistent, 100% across the board. What he saw was that in the what's called the F1 generation, I'll talk a little bit more about what that means in just a second, they were always purple. So it was almost like when he came up with this first generation, the offspring that resulted from this cross, it was almost like the characteristic of the white color in the flowers of these parent was like completely masked. It's like it almost like disappeared, almost like it wasn't even there. So he did these series of crosses to create these F1 generations, the first offspring generation, looking at these 1, 2, 3, 4, 5, 6, 7 different characteristics. And every time he saw that seemingly sort of one of these characteristics was like, and we use a word called dominant, it dominated in a sense or hid the other potential characteristic in these crosses 100% of the time. So just to kind of summarize what he saw, right, in this crossbreed. So the parent plants, these pea generations, right, green and yellow peas, while in the F1 generation, it was 100% yellow. In other words, yellow seemed to dominate or mask the expression of green. Smooth versus wrinkled, 100% were smooth. In other words, smooth seemed to dominate or mask the presence of wrinkled peas. Purple versus white. This is the one we just talked about or just maybe looked at a little bit closer. 100% of the offspring in the F1 generation were purple. Okay, short versus long stem, 100% long. So long, this long stem characteristic seemed to be the dominant characteristic. Okay, so this F, just so you guys understand where this comes from, F stands for filial, and filial means of or due from a son or daughter. So like there's this term called filial duty. Like, you guys as a son or a daughter are, have some sort of duty and responsibility to your parents, right? You guys behave and you're cool for your parents and you do stuff for them because there's some duty involved there. So it refers to sort of that relationship between sort of a parent and offspring. From the biological perspective, it denotes the generation or generations after the parental generation. So here we have the F1 generation. That's that first filial generation after this parental cross. So, and then the F2 is the second filial generation after this. So now in the F2 generation, we would take, two individuals and cross from the F1 to see what results occurred. And now here's the cool part. So let's lock in on this one right here where we have purple versus white. Okay. So Mendel, he took these purple and white flowers that were true breeding for purple and white. Generation after generation after generation, as he isolated and bred these plants, purple, 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 white, 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 white. Then he crossed them, and then all of a sudden, the F1 generation showed 100% purple offspring. Then he took individuals from the F1 generation and crossed them, okay, and ultimately it resulted in this. The results were, it was almost like the characteristic of white colored flowers had reappeared. Okay, 75% of the offspring were purple and 25% of the offspring were white colored flowers. And he saw this over and over and over again when he isolated and looked at every one of these different characteristics that he determined had this sort of idea of dominance versus recessiveness. Now we're going to get more into that, those terminologies as we go through, today and tomorrow. So, and still though, even in this F2 generation, the thing that you kind of see is that, again, 75% of them were purple. Purple, right? P-U-R-P-L-E. Okay. So you get a sense that this pattern, this this pigmentation that causes the purple colored flowers has sort of this dominance or masking effect over the white colored flower scheme, if you will. So we're going to talk more about kind of why that is, kind of as we make our way through. So another way, let's take a look at this one more time now. Okay. So here it is. Here's Mendel. He's got his true breeding plants, purple, right, and white. Okay. And he takes these true breeding plants, he crosses them, so true breeding purple flower with a true breeding white flower, and the result is 100% of the flowers were 100% of the resulting F1 generation were completely purple. Now, these things that I got drawn over here, they're called Punnett squares, and we're going to learn a little bit more about them tomorrow. But I think it's not a bad idea for us to kind of put them up here in front of you because we're going to talk more about this. And I even suggest maybe coming back to this screen after you view some of this later part of the lecture. Punnett squares, maybe you've worked with them before, are a simple way to be able to designate the potential gametes that a parent can contribute, and then the boxes on the inside of the Punnett square represents probable offspring. So a true breeding purple flower, purple is the dominant form of the allele. We use a nice big old capital P. Well, a true breeding purple flower, these are diploid cells, so they have two copies of the allele. So true breeding purple flower, its genotype would be capital P, capital P. A true breeding white flower, however, though, has two, being that it's a diploid cell, it's got two copies of the recessive allele. So that's the little p, lowercase p, and I struggle writing sometimes with my mouse. That's the little p, little p. So that's this, parent number one is here. Parent number two is here along this side, right? Now, when we do this cross in the Punnett square, we place the two different alleles, and that represents the genotype of the offspring that can result from that cross. Again, I kind of struggle doing this with my mouse, but I'm doing my best, guys. Bear with me. Okay. So now as we read this, what does this look like? All of the offspring inherited, it's 100% across the board. All of the offspring inherited one copy of an allele from this parent that is the dominant form, and one copy allele from this parent that is the recessive form. The dominant, and we're going to learn about this idea of principle of dominance. The dominant allele will mask the presence of the recessive allele. So this is why he saw in his F1 generation 100% purple flowers. Now I'm going to move my image up here, okay? Then what he did is he took the F1 generation and crossed them, or self-fertilized them, and saw what happened. So now he knows, and well, ultimately he ended up knowing based off his statistical evidence that he collected, was that he had two individuals, the two parents, okay, had, they were heterozygous, meaning that they had a dominant and a recessive allele. Same case for parent number 2, or if it's self-fertilization, the same plant, if you will. Bottom line is that these are the two resulting combinations that could result. And what do we get? Capital P, capital P, capital P, lowercase p, capital P, lowercase p, and lowercase p, lowercase p. So we use the capital letters for dominant forms of the allele. We use lowercase letters for recessive forms of the allele. What do we have here? We have this thing called the heterozygous dominant form, which has two copies of the dominant allele. Purple dominates over white, so therefore this one would be purple. Okay. I'm sorry, did I say heterozygous? I'm not sure. Homozygous dominant, homozygous dominant. We'll talk more about that in a minute. Purple. This is the heterozygous dominant form. It's got one copy. You need to have at least one copy of the dominant allele. It masks the white allele. So therefore, this one would also be purple. Same thing here. This one would also be purple, just like this one was. So what do we got? 3 out of the 4 are purple. This one has both recessive alleles. This potential combination inherited both recessive alleles. So 1 out of 4, well, 1 out of 4 is 25%. 1 out of 4 is 25%. So this represents the possibility of the flowers being white in color. So it's a 25% chance. And sure enough, what did he see? The results that he saw, right, we looked at, were across the board when he looked at these F1 to F2 generation crosses, 75% to 25% comparison, or ratio, whatever you want to call it. So ratio is probably a better word. So he starts to, work on collecting more and more data, and end up sort of, realizing, what's going on here from one generation to the next. Okay. So from these results, Mendel formulated 3 principles. So him breeding, tracking, meticulously cross-pollinating and self-pollinating, and making sure to separate plants, and repeating his experiments over and over allows him to compile a whole bunch of data. And then he ultimately starts to scratch his head, right? So he scratches his head and goes, hey, I think there's something going on here. And this ends up, he formulates sort of 3 principles. And these 3 principles, interestingly enough, like I said, 1850s or so when this is developed, are the backbone for really the rest of our conversations in this unit on genetics. So these principles describe how traits are passed from parent to offspring through sexual reproduction and meiosis. So meiosis, really when we're looking at these Punnett squares, we're sort of looking at in a sense, the concept of what meiosis is. It is this division of the potential genetic material that can be passed on to the resulting generation. Okay. So first is the principle of dominance. We've actually kind of talked about this, to believe it or not, as we've kind of gone through the first part of this lecture. Genes come in different versions that are called alleles. Okay. Diploid organisms, remember haploid versus diploid, you learned about that. Diploid organisms have a pair of alleles for each trait. Where does that pair come from? That pair comes from mom and dad. So you inherit an allele from one parent and an allele from the other parent. If present, the dominant allele is going to be expressed. So just like we talked about here, if it's present, right, all you need is one copy of the dominant allele, and that dominant form of the trait will be expressed. So in this case, the heterozygous case, they had one copy of the purple allele, so therefore the flower will be purple in color. Okay. And then the recessive allele is expressed only if the dominant allele is not present. So in other words, what do you need? You need two copies of the recessive allele in order for the that recessive form of the trait to be expressed. So this is why only 25%, okay, of these flowers were white in color, because only 25% of the time in this F2 generation were they inheriting both of the recessive alleles. It only takes one to mask the recessive allele, okay, the lowercase letter, okay? So this is kind of a repeat of this sort of concept, right? So diploid organisms inherit alleles, these Mendel's factors, right? They inherit these alleles from our parents. You know, parent number one, mom or dad, or parent number two, or mom or dad, whatever. If you inherit both of the capital letters, both of the, I should say, the dominant forms of the allele, it is said to be homozygous dominant. The prefix homo means the same. So homozygous dominant, that means the dominant form of the allele is going to be shown, and by having two copies, the dominant form of the allele is being shown. Let's go over to homozygous recessive here, okay? Having two copies of the recessive allele inherited from mom and dad. Homo meaning the same again, recessive allele. So having both copies of the recessive allele results in the recessive form of the trait being expressed. So the white color of the plant, or the flowers in our kind of running example that we've been using. And then kind of smacked in the middle is, you see, again, like we talked about, having the capital letter and the lowercase letter, having one copy of the dominant form of the allele and one copy of the recessive form of the allele. This is said to be the heterozygous individual. And typically, at least for our purposes right now, we're going to talk about it as, and I'm going to use the wording heterozygous dominant, okay? Because there are sometimes these heterozygous cases that are a little funky later on, and we'll get to that later on. But in sort of typical Mendelian genetics and his pea plants, we say heterozygous dominant. Again, what does that mean? All you need is one copy of the dominant allele, and that is going to ultimately express the dominant form of the trait. So in the purple versus white colored flower example we've been running along with here, you get one copy of it, it's going to be purple colored flowers. Okay. Second, principle of segregation. Diploid organisms inherit a pair of alleles for each trait, one from each parent. So the two alleles segregate from each other during meiosis so that gametes, egg and sperm, will end up with one allele or another, not both. So this sounds, to me, I always think about this sounds more complex than really what it is. It kind of goes back to this, right? You know, think about it from this perspective. You know, your mom has two forms of alleles that express the traits that they possess. Now, when sex cells are created by the parents, right, those sex cells can only have, because we learned about in meiosis, you go from diploid cells down to haploid cells, right? So they can only pass on one or the other. Same with, dad, right? They can only pass on one or the other form of the allele. So I think this next little graphic will kind of help you guys sort of understand. So here's diploid parent that has these, here's the capital S versus little lowercase s, right? And they got it color coded with red and blue. So when they go through meiosis, right, you know, each chromosome has to be duplicated. So now we have these duplicated sides with the sister chromatids attached, right? And then ultimately, as meiosis goes through, it gets divided, right? Again, and the result is that the gametes that are going to be passed along can only result in being given, you know, 2 out of the 4 resulting cells have the dominant allele, and 2 of the resulting cells have the recessive allele. 2 of the 4 resulting cells have the recessive allele. In other words, right, parent only has, the parent cell or the parent individual or plant or whatever it is, has the dominant and recessive allele in this case. Well, they can't pass on both to their offspring because they can't pass on both to the sex cells that they produce. So that results in then ultimately haploid cells that are going to either have one or the other. And it kind of goes back to these Punnett squares, to be honest with you. That's the whole concept. Each side represents a parent, right? A parent. And each parent can give or pass on only one or the other allele. And then the inside of this Punnett square represents the probability of the offspring that can outcome from that cross. It's a probability, right? Peek that a little bit in your mind a little bit. Think about it. It's a probability, okay? It's not an absolute guarantee, and we'll have some more discussion about that as we kind of go through this unit. All right, boom, boom, boom. Good, we're back to here. Sorry, flipping some screens there for a second. All right, so that is the principle of segregation. Just the idea that, you know, when 2 alleles segregate during meiosis, they will only end up with one allele or another, but not both. And the interesting part about it is, like, there are situations in some, and we'll talk about some of these genetic disorders, that in the process of meiosis, if segregation doesn't take place correctly, the result can be inconsistent division of chromosomes. And sometimes this process can get messed up. And it can result in sex cells that can carry one too many chromosomes or even not enough chromosomes. So that's a story to be kind of talked about later. But you guys know me, I sometimes like tangents, sorry. And I think it's good to foreshadow some of the things that we'll end up talking about. All right, last for today, and we'll finish it up and get you guys out of here. The principle of independent assortment. Again, this one sometimes to me maybe sounds a little wordy to an extent, but I'm going to try to sort of hopefully simplify it as best I can. So how the chromosomes how the chromosomes in each homologous pair line up during metaphase 1 is random. So metaphase 1 here, right? So metaphase 1, it's randomized, right? So they've got, here's this cell that's got 4 total chromosomes. As it's going to be going into metaphase 1, it could end up aligning themselves in one of 2 ways, 2 possible combinations, if you will. Okay. So the directionality that these, if we got all blues on this side and all reds on this side, and then we got a blue and a red and a red and a blue, okay. The reality of it is that we have to talk about it from an idea of probability. It's randomized, okay. So how the chromosomes line in metaphase 1 is random, and maternal versus paternal. Maternal refers to from each one of the parents. Maternal refers to the mother, right? And paternal, the father. I had to think for a second there. My vocabulary is being tested again. And then these maternal and paternal homologs can face either pole in each pair. Thus, the alleles for different traits get matched together in all possible combinations. Right, so to make a long story short, okay, the probability of the outcomes as sex cells are created is a probability of having to look at it from the perspective of all possible combinations. Let's just go to the next slide. I think I'm going to kind of talk you through this and help this sound a little bit better. So in this example that we just looked at, there's something very similar to this in your textbook in chapter 10. I would recommend taking a look at it. As you're looking at this, pause the video. But here's the deal. So this individual, this organism shows it's heterozygous organism that's diploid for a plant with round and yellow seeds, or pea plants of Mendel, right? It possesses two different. Boom, boom, boom. I'm sorry, I'm stumbling a little bit. This is a diploid cell, right? So a diploid cell has 4 total chromosomes, okay? The haploid individual, or the haploid cells, if the diploid number, I should say, we learned about this in meiosis last week. If the diploid number is 4, right? Okay, then the haploid number is going to be 2. Okay, so each one of these sex cells, or these gametes that are going to be produced, need to have 2 sets of chromosomes in them. Okay, so this value, the N value that you learned about, is the haploid number. Okay, so we place that in here in this little calculation that we just talked about. The number of possible combinations is 2 to the nth power, or the n power. N represents the number of chromosomes in a diploid cell. On this simple little example, it has, okay, a diploid value of 2. So 2 to the second is 4. There are 4 resulting possible combinations that you could come up with. 1 here, big R, little y, big R, little y. This is the second possible combination. Little r, big Y, little r, big Y. This is the third possible combination. Little r, little y, little r, little y. And this is the fourth possible combination. Big R, big Y, big R, big Y. So 4 possible combinations. Again, recognize these sets, they're identical, right? I mean, you learned about that in meiosis last week, right? That these 2 cells are identical to one another in terms of their distribution of the alleles. So 1, 2, 3, 4 possible combinations. Now here's kind of cool. Humans, right? Humans as diploid organisms, we have 46 chromosomes. When sex cells are produced in humans as haploid cells, that means we have 46 total chromosomes, 23 pairs of chromosomes, 1 inherited from mom, 1 inherited from dad. That means the haploid value for us, the N value, right, is 23. So 23 for the N value. So what's the significance of that, right? So 2 to the 23rd power. What's 2 to the 23rd power, right? Let's bring up my calculator here. It's kind of cool. 2 to the 23rd power. Okay. So what does that look like? That's almost 8.4 million different combinations that can result in the production of human sex cells. So for those of you that have brothers and sisters, right? I got 2 older brothers, and while myself and my 2 older brothers do look similar to one another, and yes, you could probably stand the 3 of us next to us in a room and you'd go, yep, I see how that's Mr. P's older brothers and they're related. However, and we obviously both have the same parents. The reality of it is that we are not identical, okay? And partly because of this exact concept here, right? There are almost 8.4 million possible combinations that are produced in human sex cells. Okay. In addition, you guys learned about in meiosis last week about crossing over and recombination. It's so doubly this sort of shuffling of the deck concept. There's nearly 8.4 million possible combinations that could result in sex cells that are produced by your parents, that ultimately resulted in you. But on top of that, you take that combination, and those combinations the deck gets shuffled, right? By crossing over and recombination, crossing over and recombination. So you can see, that when you start to look at organisms that have, many more chromosomes, much more genetic information, the variability and complexity of the resulting number of combinations can be pretty crazy huge. But what was that? My kids are running around upstairs. But the basics of it revolves around what Mendel gave to us in the 1850s. All right, so hopefully you guys are doing okay with this. Again, I would suggest that if there's questions about this or whatever, go back. You can always go back and review things. Take a look at those sections in your book, 10.1 and 10.2. Read them carefully to be able to help you through this information. I will see you guys again tomorrow in our second kind of section of this video lecture, mainly looking at Punnett squares and pedigree charts a little further to set up how those are worked and how those are used. And we'll look at what we looked at today, just so you guys know. These Punnett squares we looked at, these were all what are called monohybrid crosses. They look at only one particular trait. Tomorrow, we'll look at dihybrid crosses as well, too, in these Punnett squares that look at the combination of two different traits. So the Punnett squares are going to get a little bit bigger. And then we'll talk about what these things called pedigree charts are. They're used by geneticists as a way to kind of predict outcomes or to be able to track sometimes different genetic disorders within families. All right. Anyway, that's it. Thanks, guys, for listening. I'll see you tomorrow on tomorrow's video lecture. Good job. Keep it up. Again, don't forget, follow at Whiteboard Mr. P on Twitter if you haven't already. And that is it. Good day.